NASA's Jet Propulsion Laboratory presents the Von Karman Lecture, a series of talks by scientists and engineers who are exploring our planet, our solar system, and all that lies beyond.
Hey, good evening, everybody. How's everyone tonight? Superb. Again, as always, thank you very much for coming out to join us tonight. So, the search for life elsewhere in the solar system has tantalized humanity for centuries. And as much as this search has led us to look outward towards places that may have life or the chemical precursors for life, it has also led us inward, studying places on Earth and recreating other worlds in the laboratory that can act as analog environments to other places that are far more difficult to reach. Titan, a moon of Saturn, is an excellent example of a prebiotic world where a diverse array of organic molecules exist, but life as we know it cannot survive on the surface. The liquid hydrocarbon lakes of Titan are a unique environment where organic molecules have the opportunity to interact and possibly react with each other. By recreating Titan's lakes in the laboratory, we are discovering new chemical interactions that were previously unknown to science and which may help us understand how Titan came to be. Iceland, a Nordic island shaped by volcanism and glaciers, is recognized as an analog environment for Mars due to its similar geochemistry and mineralogy. 2 field expeditions there have successfully tested life detection techniques that may be used on future Mars missions, discovering that microbial diversity can vary widely, even in areas that appear to be the same in terms of geology. This has implications for where and how we might search for life on Mars. Tonight's guest is a technologist in the Instrument Systems Implementation and Concepts section right here at JPL. She's also the Assistant Project Science Systems Engineer on the Cassini mission, which has been orbiting Saturn for the last 10 years. Her research focuses on organic and biomarker detection strategies through both in situ and remote sensing techniques. While earning her PhD in chemistry at the California Institute of Technology, she designed receptor sites for the detection of bacterial spores, the toughest form of life. As a NASA postdoctoral fellow right here at JPL, she developed novel protocols to analyze organics, such as fatty acids, using small, portable microfluidic sensors. She is currently working as a collaborator on the Mapping Imaging Spectrometer for Europa, an instrument selected for NASA's next mission to Jupiter's icy moon Europa. This spectrometer will map Europa's surface and search for organics, salts, and minerals. Ladies and gentlemen, please help me welcome tonight's guest, Dr. Morgan Cable. Get him, girl.
Hello. Can you guys hear me all right? All right. Thank you so much for coming tonight. I'm very excited to be here, to have you guys be here to tell you a little bit about some analog environments that we use to study the solar system. So first, I wanted to ask you a question. So analog environments, we're looking at places on Earth or places that we can simulate in the lab that may replicate places elsewhere in the solar system. So is this Mars or Earth? Yeah, this one was a little easy. There are plants in here. But this is Jordan. This is actually where The Martian was filmed. Okay, so how about this one? Good. Okay, you guys are doing pretty well. What about this? Are you sure? You're right, this is in Libya. How about this? You guys are really good. I was hoping I would trick you. This is Salisbury Peak. Okay, what about this last one? Oh, not so sure now, are you? This is Iceland. Yeah, so many environments on Earth, places like Jordan and Libya, the Atacama Desert, have been used by NASA as analog environments for places like Mars for a while, and Iceland is one of these places. It was actually used in the 50s and 60s to train Apollo astronauts for some of their activities on the moon. The reason that we like it as an analog for Mars is because it's very volcanically active, right? It's a land of fire and ice, glaciers and volcanism. A lot of those volcanic regions are very similar in some of their properties to places on Mars. It's also typically has areas that are desiccated, so not a lot of water, low nutrient availability, just like Mars, temperature extremes, and also some isolation from us from contamination that could fool some of our life detection techniques. So we really like Iceland as an analog for Mars. Now I'm part of a team, and it's a NASA team, so of course we love our acronyms. Feldspar is a type of volcanic mineral, and that's what we decided to name our team, which stands for, I'll only say it one time, I promise, Field Exploration and Life Detection Sampling for Planetary and Astrobiology Research. Yes, Feldspar, go team Feldspar. Okay, so this is the team, and this is all the places that we're from. And I wanted to take a minute and describe how we all met, because this was a very unique set of circumstances. You can tell we're all from a variety of different places, both in the US and over in Europe. And we met through something called the NASA Nordic Astrobiology Summer School. Raise your hand if you've heard of this before. Okay, you should all Google this, because this is an amazing program. It's meant for early career scientists and engineers, people interested in learning about astrobiology. And this, wait, hang on, I have a laser pointer. Okay. This gentleman right here, Wolf Geppert, organizes the one that's in Iceland. It alternates between Iceland and Hawaii. It's every, I think, year and a half, and it's a wonderful program. So as a chemist, my background is in chemistry, I went and learned about geology and biology and geophysics, things that are important for the study of astrobiology. It's a very interdisciplinary field. And so the biologists would learn some chemistry, and the geologists would too, and it was a great way for us to interact. And so we went around and toured a few lava fields and some glaciers and other environments on Iceland that are good analogs for planetary environments. And a few of us were thinking, boy, these are some really cool places. And I haven't found much in the literature of people actually using these environments to test things for Mars. Maybe we should do something about that. And so this smaller group, I think there are 50 that were in the astrobiology summer school, but a group of about 12 or 13 of us decided to cobble together funding from whatever sources we could get and put together an expedition. So after that summer school, the following year, we were able to come back to Iceland and do our own smaller expedition and start our question to inform future Mars missions that are looking for life. So this is not Mars. Again, this is Iceland, so ignore the beautiful Icelandic glaciers in the background and pretend like you're on Mars, okay? You've landed your rover. So there it is. And you've used high-resolution imagery. This is actually an image of the Mars Curiosity rover. Can you see it? This is from Mars. It's right there in the middle, taken by HiRISE, which is a camera aboard the Mars Reconnaissance Orbiter. So you've used your high-resolution imagery to pick your spot that you think has life, all right? And you've landed your rover, and you've got all sorts of instruments, including your Life Finder 3000. This is a future Mars lander, Mars rover, and you're ready to look for life. Okay, well, where do you look? Do you look here? Do you look over here? In terms of the really close, small context, this is the best imaging you have, is what's coming from your rover. And who knows, the alien could be hiding right behind that rock that you don't sample. So our question was, when you get to this place and it looks, the technical term would be homogeneous. So it sort of all looks the same. If you drive your rover over here and you collect a sample, and then you drive your rover over here and collect another sample, will they be the same? Would you find life in one and not the other? And how do you decide? So that was a question that we were trying to answer. And so we have Mars on the left, Earth on the right. So a typical Mars type exploration for this, again, you'd start with that remote sensing. We don't have MRO orbiting around Earth, so we used Google Earth to do some of our very high resolution imagery of our site. So this is one of the lava fields that we visited. Now, we didn't have a Mars rover, but we had a land rover, which is what we used to go and visit our various sample sites. We tried to pick places that were sort of off the beaten path. Even in Iceland, it's become quite a tourist site, and so we're trying to find places where we can find fresh lava, but that doesn't have a lot of tourist traffic. And just like we do on Mars with the Curiosity rover, which has several techniques for doing standoff analysis, things like the tunable laser spectrometer, TLS, or laser ablation types of experiments. We also use some field portable instruments. This is me with what's called an ASD spectrometer, which is sort of strapped here, and we have a probe that we use that we point at samples. And this instrument looks at the light that's reflected, that comes from the sun, that's reflected off the rocks and comes back. And you can identify minerals this way. And it's very similar to some instruments that we've included on Mars rovers in the past. So now once you've collected your sample, a typical Mars rover will bring it inside and interrogate it with a variety of different instruments. Well, we have a field lab, and a bunch of my colleagues, this is my friend Elena, who's studying, she's getting her PhD up at the University of Washington. And so she pretended to be one of the instruments. Okay, so we're doing this, sort of thinking about what would a Mars rover do, and now what would we do? And there are plenty of places that we could go on Iceland. It's one of the most volcanically active areas on Earth. It's growing, it's sort of sandwiched between two plates, and one of those plates is growing, I think, at 5 centimeters per year, which is, in fact, is the same rate as your fingernails grow. Kind of cool. Okay, so these are the different places that we decided to look in order of age. All right, now, raise your hand if you're Icelandic. Oh, okay, so I apologize if I butcher any of the Icelandic. This is Melifellssandur, which is the oldest site. This is sort of a volcanic desert, kind of in the middle of nowhere in Iceland, which is good, so not a lot of people go there. This is our oldest site. This is on the order of about 10,000 years. Landmannalaugar, which is in the Central Highlands. This is from an eruption in the 1400s. Eldfell, which is actually a relatively new eruption on this tiny little island called Jaime. This eruption happened in 1973, and that's more of a lava fissure. And then our newest site is Fimmvörðuháls, which is a new lava field. Do you guys remember back in 2010, the air traffic was really messed up in Europe? That was because of this. And the glacier that it erupted from underneath is called Eyjafjallajökull. Don't ask me to say it again. I've been practicing. Okay, so, but I'm only going to be talking about these two today. So this is what the Jaime eruption looked like. You can see a couple of the other islands that are in this island chain down off the southern coast of Iceland. It's actually funny, you see that little white dot? That's somebody's house. He's the only guy that lives on that island. There's one house. But this eruption happened sort of on the eastern side of the island, and the island actually grew by about 25% over a period of a few months as lava poured out. This is a tiny island. It's a fishing town, basically, and the lava almost encroached into the bay where, you know, all the harbor where the fishing boats were, and they were really concerned that it was going to block it off completely and they would lose their livelihood. So they brought in these boats, you know, the fire boats that shoot water, and we're doing that for months and months and months, and we're able to stop the flow of lava. So now they have a more protected harbor because of this eruption. Well, this was 1973, and this is the more recent Fimmvörðuháls Eyjafjallajökull twice. Yes, okay, eruption. And so these are the two that I'll be talking about today. Now, we didn't go to them when they looked like this. When we went, they looked like this. It's a little dark because it was a little cloudy when we were there. But let me see if I can play this. So we also brought with us a quadcopter to get some more context imaging. And you can see it's a little bit dim, but beautiful landscape, a lot of ice and a lot of lava, and not a lot of magma, right? So now it's cooled and it's had time for life to gain a foothold, and that's what we're interested in. We're interested in how does life gain a foothold? Where does it start to colonize first? What types of life are there? Okay, so at these sampling sites, we were thinking like a rover, but we're also thinking like a sampling team here on Earth. Now, rovers have a very small set of samples that they can collect, right? Every single sample is precious. But for us, with our team, we could collect many samples and then go back and do that thought exercise of, okay, would that have been the one that we would have picked? And so what we decided to do was collect samples. We do it in triplicate, which means we collect 3 in the same place at 5 centimeters depth at 1 meters spacing. So we collect 3 here and then go 1 meter away and do it again. And then we'd go 10 meters away, do it again, and then 100 meters away and do it again, so that we could get an idea of scale, right? So orders of magnitude. And we used as sterile of technique as we could. So we sterilized our shovels and trowels before we collected the sample. We approached from downwind, all those things to try to minimize anything from us contaminating this fresh lava field that we were trying to collect samples from, to be sure that we didn't contaminate anything. Okay, so here's an example of one of our sampling areas. So you can see we collected 3 from each. And also, after we were done collecting the samples, we would go in with our standoff techniques and get some additional data on the mineralogy too. Once we collected the samples, we brought them back to our field lab. This is in a tiny town called Kvosfellur, which the headmistress was kind enough to let us use their school as our field lab because the students were out for the summer. This is in the middle of July, so the sun never fully set while we were there. So we could work all day because there was no night. But we loved it, and it really was a beautiful time to be in Iceland. So for the biology type experiments, again, we used the sterile technique, but we split up the samples, and for some of them, we would grind them and then look at them with some other mineralogy techniques. This is a Raman spectrometer. And so what did we look for? Well, one experiment that we did was called PCR. PCR stands for polymerase chain reaction. It's basically a fancy way of saying that you pick a certain type of DNA, an organism that you're interested in looking for, and you make a lot of copies of its DNA until you have enough that you can actually detect it and quantify it also. And we looked for bacteria, fungi, and archaea, which are the 3 sort of main kingdoms in the tree of life. And we were interested in seeing how much of each of these were present, and if that varied depending on where we are or where we were sampling. So this is our fresh lava field. This is the one that messed up the air traffic. I'm just going to call it FIM for short. And so this is an example of that collection scheme I talked about, where for every green dot, that's a 1 sample site. So you've got ones that are 1 meter apart, 10 meters apart, and 100 meters apart. Now, we don't really need the numbers here, so I'm going to nix those for now. But what I'm going to do is take that green dot, and I'm going to expand it to the size of how much bacteria were present, how much bacteria we found. As soon as I hit the forward button. There we go. Okay, now I said that we collected things in triplicate. So there are 3 circles for each sample site because we analyzed that 3 times. And what you can see is that this is the amount of DNA of bacteria by size. So this is a lot, this is a little. You can see even on like 1 meter differences, the amount of bacteria that we see there varies a lot, way more than we expected. So that means if you were standing in one spot and you went 3 feet over, you would see a big difference in the amount of life that was there. Now, this means that it's very, very important where you're looking for life, that you have to be very clear on where you're sampling to be sure that you can try to detect life and not miss it. So we were surprised by that. And even in the samples that we collected, 3 samples in the same spot, we would have a lot of variation. Now, this wasn't true with just bacteria. Here is fungi. And again, you can see even on the 1 meter difference, 1 meter spacing, you can see huge differences in the amount of life that was there. We find fungi everywhere. They're a lichen that are a type of fungus in symbiotic relationship with cyanobacteria that live in Antarctica and can survive Mars conditions. We see them all over the place. And it was interesting to find them here in this very new lava field. But we were, again, surprised with how much variability there was just between these different samples that we collected. And archaea was the same. We didn't find quite as much archaea. Archaea are, they're sort of the most primitive form of life. Unlike bacteria, they don't have mitochondria inside of their cells. And we, again, find these everywhere, too. They're really, really robust. And we saw this incredible variability. So how does that compare to our other lava field? Remember, I mentioned I was going to be talking about 2. So this is our younger one, and this is the older one. Still not old in geology terms. Geologists think about things on like millions of years, but it's old in terms of the life that we were finding. And we do see differences. Now I'm going to add sort of error bars to these. So these blue sort of circles represent the errors in the measurements. What I did was I averaged every single sample that we took from FIM of the bacteria, and that's this one here. And everything from Eldfell, the island sample, and that's here. And you can see for the older lava field, the error bars, the The blue rings are smaller, they're thinner. So these were more reproducible in terms of the amount of life that we found, whereas things kind of varied much more for the younger lava field, which is very interesting to us. So maybe this means that it takes a while for life to stabilize, that things vary quite a bit. They're more heterogeneous at first, but then they become more homogeneous over time. Now, that was one technique that we used. Another technique that we looked at was ATP. Now, ATP stands for adenosine triphosphate. It is the energy molecule that all life that we know of, uses. And it also doesn't hang around very long in the environment once a cell dies. So it's a really good way of telling you not just what organisms are there. It can't tell you identify specific organisms because everybody uses it. It's sort of like your energy. But it can tell you whether or not they're alive, like right now. They're actively metabolizing, like us. In 2013, now we're looking at a bar graph, but I've sort of highlighted things that differ on a 10-meter scale. So these guys differ in a meter, these guys differ in a meter, but these and these are 10 meters spaced apart. And you can see that there's a lot of variability. Even things that were right next to each other vary widely in the amount of energy, the amount of life that's active, that's present there. But in 2015, now this is the same lava field, this is that young lava field, now we start to see some patterns emerging, right? This grouping is all at one spatial scale of 1 meter apart, and it's starting to look distinctly different from this grouping and this one. So it seems that over time, we're starting to get more predictability, at least, and less variation on these distance scales between these samples. Oh, and I forgot to mention, okay, so look at the scale here. This goes up to 3,000. This goes up to 10,000. So let's make them the same. You'll also notice that life is increasing, which makes sense, right? That we're over time starting to build up a stronger community, more diverse, hopefully, microbial community here. So this is something that we're hoping to monitor over time. We have 2 data points in terms of 2 expeditions that we've been to visit and sample this place, and we're hoping to go back. So to conclude, when it comes to things like this, there's no real ending to the story, at least not yet. We're just starting on this. We've done 2 expeditions. We just got funding to do 4 more over the next 4 years, which is really exciting. So we're going to go back to these lava fields that I told you about, and also a new one. This one is called, it's up in the northern region of Iceland, which is called Bardarbunga, and this particular lava field is called Nornathraun, which means witch's hair in Icelandic, which is pretty cool. But this one erupted in 2014, and we've gotten permission to be able to go up there. And so this one will get even more of a head start at trying to see exactly where life starts, what starts first, and then what comes later. So some conclusions that we've drawn just from our limited data so far. We know that the older lava fields have different types of life than the younger lava fields. In the younger ones, we also see that life abundance varies widely, even at spatial scales of just a meter away. But it does appear, at least tentatively, that over time, patterns do emerge. So our plan is to continue to go back and monitor this emergence of life in fresh lava fields over time. So that's the fire and ice part. Now let's talk about the methane-ethane part. So this is part 2. For Mars, we have analogues on Earth. But there are some places in the universe, or in the solar system at least, that are so weird, we don't have any analogues here on Earth. I'd like to introduce you to Titan. This is a moon around Saturn. It's actually the largest moon in the solar system if you count its atmosphere. Yes, it has an atmosphere. It's actually thicker than ours, about 1.5 times thicker. Once you strip that atmosphere away, though, then it's a little bit smaller than Ganymede, but second place, that's all right. It's got an atmosphere that's about 96% nitrogen, a little bit of methane, and it's thicker, like I mentioned, than ours. The surface is this mantle of water ice. Water ice takes the form of rock on Titan. It's the hardness of granite at these temperatures, because it's way, way out past the sun, or far away from the sun, so it's much colder. And so you have this mantle of water ice, and it's coated in this veneer of organic molecules. We've got dunes that are also, we think, made of those organic molecules. And then we've got this hydrologic cycle. But by hydro, I don't mean water. I mean hydrocarbon, methane and ethane. This is an example of a specular reflection. You know, if you're in an airplane and you sort of get that sun glint off of liquid water, that's what you're seeing here. This is off of a lake that's in the northern pole of Titan that's made out of mostly methane. So we have methane and ethane that form clouds and rain down onto the surface, and we may even have some cryovolcanoes, although this is still hotly debated in the Titan community. So we may have evidence where, okay, so Titan, just like Europa and Enceladus and all these other icy moons, has an ocean of water. Underneath all this stuff, like deeper down, ocean of liquid water. And in some places, for example, Sotra Facula, we may see some of that liquid water maybe mixed with some stuff like methanol coming up to the surface, maybe, which would be really exciting because then more chemistry could happen. So Titan's a fascinating place. And it is similar to Earth in some ways, right? It has clouds. Earth has clouds. They're made out of different stuff, but they both have clouds and atmospheres and those types of behaviors. They both have river channels. They are moving different things in those channels, but in terms of their geology, they do look very similar. They have lakes. This is Ligeia Mare, which is in the North Pole of Titan. Its surface area is actually twice as big as Lake Michigan, so it's a big lake. And we've got remnants of ridges, other sort of geological features on Earth we see echoed in Titan as well. The scales may be a little bit different, but we still see very similar behavior. So maybe that suggests similar processes, even though they're made of very different things. And the dunes, which are crazy cool, they're hundreds of kilometers long, I think like 10 kilometers high. Mike, is that right? 10 kilometers-ish? Yeah. We're not sure if they're moving or if they're frozen dunes, but we believe that they're formed by organic chemistry happening in the atmosphere. So the atmosphere has just two things in it, nitrogen and methane, until you introduce energy. Now the energy can be UV radiation from the sun. Even though it's far away, over time, that can add up and cause chemistry. You have these really fast-moving protons and electrons that are accelerated by Saturn that go slamming into these molecules and they break them apart. And so pretty much any combination of carbon, hydrogen, and nitrogen that you can think of, that you can make in your little chemistry set at home. Do you guys have chemistry sets at home? Anything that you can make probably exists on Titan. You start to form these complex molecules that continue to react and sort of combine with each other as they form these haze layers that we see in the atmosphere, and then eventually they make it down to the surface. And we think that's what is making up these dunes, in addition to other surface features on Titan. Okay, so let's go back to the lakes, because, you know, if it looks like a duck, but in this case, it's not actually looking like a duck. I mean, we do see some things. We see bathymetry. It has similar depth, similar properties in terms of appearance to Earth lakes, but they're not, right? These are filled with something very different, methane, ethane, a little bit of propane. So Earth, we don't have a good analog, unless we were to go and dump a lot of propane in a lake, and I don't think anyone would be too happy with us if we did that. And plus, it would boil away because it's too hot here. So what do? Well, we go to the lab, and we can make mini Titan lakes in the lab using things called cryostats. So we use liquid nitrogen, which is actually a little bit colder than Titan's surface. Titan's surface is about 90 Kelvin, which is minus 183 degrees C, and I don't know what it is in Fahrenheit. I should know that, but it's cold. Liquid nitrogen is even colder. It's about 77, 78 Kelvin. So we use that as our bath, and then we heat it up just a little bit, about 10 or 12 degrees to get up to Titan temperatures. This is what it looks like. So we've got our doer. That's the silver thing here. It keeps hot things hot. In our case, it keeps cold things cold. We put our liquid nitrogen in here, and then inside of this beaker within a beaker is a little Titan lake that's liquid ethane in there. And so this is what we'll use to do our experiments. Now, there are a lot of fascinating things going on Titan. We've got atmospheric chemistry. We've got rain, methane and ethane rains down on the surface. It may be dissolving some things or potentially forming caves or other interesting types of geologic features. It may be flowing things into these lakes. Stuff that gets dissolved can move into those lakes. And then the lakes themselves, they can evaporate over time. And so we have these really interesting cycles that we would like to study and understand better. So let's take them one at a time, and I'm not going to do them all, I'm just doing 2. But let's look first at soluble materials dissolution. Basically, what dissolves in a Titan lake. Okay, this is a setup from one of my colleagues, Mike Malaska, put together to try to answer that question. So again, we've got our doer, which keeps hot things hot or cold things cold, and a lot of liquid nitrogen. Now, if you look at a schematic of this is a figure from one of Mike's presentations. We'll fill it with liquid methane or liquid ethane, and then this is our probe that we'll use to measure concentrations of things that we dissolve in there that we're interested in studying to see how well they dissolve in this liquid. And so we'll do what's called a flush and fill to sort of push things out, and it has to come back through a filter before it comes back in. So we don't want any chunks. You know, if you dissolve something like sugar in your coffee and you put too much sugar in there and some of it kind of falls to the bottom, we don't want that to interfere with our measurements. So we have a filter. So only the things that are actually dissolved in the liquid are what we'll measure. Now, a few of the things that Mike has measured so far are these organic molecules here, benzene, naphthalene, and biphenyl. And the amounts that dissolve in a Titan lake range from about 18 milligrams per liter down to much lower than that. Now, to put this in context, sugar dissolving in water at room temperature, any guesses on how many mgs per liter that is? It's way more than 18. It's almost 2 million, actually. So not a lot of this dissolves in methane or ethane. But this is still interesting for us to try to understand, right? Because it's cold, because it's made out of methane and ethane instead of water. Water's polar, these things aren't, so they behave differently. And this helps us understand and generate models that we can start to think about processes that have happened on Titan over very long timescales, millions or even billions of years. And this can help us understand what types of processes or physical landforms we see on the surface, if these are due to certain things dissolving and certain things being left behind, for example. And it also, these experiments can tell us about some interesting features that we've seen. For example, there's something that was observed by the Cassini mission happening over a period of many years. We noticed that this feature, this is again Ligeia Mare, a lake at the northern pole of Titan. We saw this bright feature appear in, this is in radar data, synthetic aperture radar data. And we see it appear and then disappear over time. And this is called a magic island. Jason Hofgartner wrote about this recently, and I saw him, he's here somewhere. There he is. And so this was really interesting, right? What is this? It's a transient feature, right? It comes and goes. So is it something floating on methane and ethane? It can't be methane or ethane ice. Unlike water ice, that ice sinks. It doesn't float. So it can't be that. Could it be something else? We don't know, but we have a theory. Based on some of the experiments that we've been doing, our theory is that if you have a lake that's rich in ethane, and then you get a rainfall, nitrogen, which is in the atmosphere, dissolves a little bit in both of these, but it's much more soluble in methane than it is in ethane. And so when this nitrogen-saturated methane hits this lake, it can cause some of that nitrogen to boil off. It's called exsolvate, but it just means bubbles happen. And it can generate something. Maybe that could be an explanation for these magic islands. And Mike actually did an experiment in the lab where it looks like something similar could be happening. So what you're looking at here, this is a glass container. There's a stir bar that's moving around in here. On the outside is a bath of methane that has nitrogen dissolved in it. And on the inside, he's trying to condense some ethane. Now, when that happens, you generate a little bit of heat, just a little bit. And it's just enough that now you can start to see these bubbles. Do you see these bubbles happening? That's the nitrogen that's boiling out, that's exsolvating out. So we see some things in the laboratory that maybe we can use to attribute to physical processes that are happening on Titan. All right, so let's look at one more process that may be going on Titan. We're interested in these lakes and what can happen when the volume of those lakes changes. So what happens when things evaporate from a Titan lake? Well, we have some evidence that something interesting is going on from the Cassini mission. We've noticed that around some of these lakes, we'll see what we sort of call or nickname bathtub rings. They sort of look like evaporitic deposits. that are happening perhaps as the lake is evaporating away. We don't know if that's actually the process, that's just a guess, but it is interesting. And we started to think, okay, well, what could these be made up of? Well, let's go back to that solubility experiment Mike did. Well, benzene is not very soluble, so if the lake liquid volume changes because of evaporation and it's dissolved to what we call to saturation. So you put in as much benzene in that liquid as you possibly can. If some of it evaporates away, the liquid, some of that benzene is going to crash out and precipitate out. And so this could be something that's happening. And so we decided to do an experiment in the lab to simulate this. So what you're looking at is this is a zoomed in microscopic image. This is a droplet of benzene, so that you're looking at the edge of it. So this is benzene up here that's frozen, because it's definitely a solid at Titan temperatures. And this is in liquid ethane. So you can imagine that this is sort of like if an ethane lake started to evaporate away and some of the benzene was left behind. Now what we noticed was when we mixed these two together and we waited, we saw something interesting. This is something called recrystallization. You can see how there are some crystals, solid chunks here, but then over here, they've broken up into tiny little bits. And we didn't do anything. We didn't touch the sample. We just sat it there and watched this happen. And we saw this happen over and over again. Now, in chemistry, we get very excited when something like this happens because it tells us that something is changing inside of the sample. Whether it's a physical or a chemical change, it's a change. And it's at really cold temperatures, which is exciting. Normally, things slow down or even stop at very low temperatures. So this was something we wanted to investigate further. So we turned to one of our instruments. This is data from a Raman spectrometer. And essentially what you're looking at is peaks that are attributed to different molecules that are present in our sample. Now we just have two things in there, right? We've got our benzene and we've got our ethane. And what we noticed is that once you mix them and you wait for a little bit, we see this new peak appear over here. And we also see some shifts in these peaks over here. Now this tells us, this particular technique, not only does it tell you what's there, but it tells you the chemical environment that it's in as well. Now this peak growing in, this is one of the ethane peaks. This tells us that some population of the ethane molecules that are in our sample are in a different environment than the rest of the liquid. So we started to try to think of what that different environment could be. Well, we've got two things. We've got benzene and ethane. How can you combine them or mix them together to sort of trap ethane in some kind of like a co-crystal or some other sort of state? Well, we turn to the literature. And there's a paper on a similar molecular pair, same with benzene, but now we've got a different molecule in the middle called acetylene. But they form this really cool sort of sandwich structure where the acetylene was stabilized inside. And so we thought, okay, well, ethane's very similar to acetylene. It's just got a few extra protons, so maybe this is what's going on. We did some modeling that supported this as well. But then we decided, well, we've got this colleague in Australia who has access to their synchrotron. So let's go down there and do some cryogenic experiments. And so one of our colleagues went down there and basically fired a bunch of X-rays at our sample, kept it cold, and when X-rays bounce off of a crystal structure, that pattern of how the rays are bounced off and diffracted can tell you what the atoms are and where they are, what their specific arrangement is. So this can tell you what the crystal structure is. It's like being able to zoom in and look at individual atoms, which is very cool. And so we did that, and this is what we saw. Very, very different from what we expected. So you can see there's like a ring of benzenes, and then it's kind of hard to see, but you've basically got this line of ethane molecules that are forming sort of like a tube. Totally not what we expected. Really wicked cool. We were very excited. So if you sort of like drape a cloth over the benzenes, you can sort of get a better idea of the structure. We call this an inclusion compound. And ethanes form in these channels in between the benzenes. Really unusual. No one had ever seen this before we did this experiment. And this was really exciting for us, both from a chemistry perspective and from a what the heck is going on Titan perspective, because this helps give us some insight into weird processes that could be happening on the surface. So Titan's surface landscape is strongly influenced by this complex organic cycle, and we're starting to get some insights through doing lab experiments to try to understand what's going on. We know that certain molecules dissolve, and thanks to the work from some of our colleagues, we now know exactly how much dissolves. We know that some of them precipitate or reorganize, like that cool crystal structure I just showed you. And this leads to some novel, solid geological materials. This is the equivalent of like a hydrated mineral on Earth. And so this can help us try to understand how things are stored, how they move, how they interact on this really exotic alien world. But of course, this is just part of the story. Again, doesn't have a full ending yet. We definitely need more lab work. We need future missions to go to this amazing moon. Cassini has given us an incredible wealth of data, but it's also given us more questions than answers, which as scientists is what we love. So I'm hoping that we'll be able to go back and study Titan further in the future to learn about what makes it tick. So with that, I have a lot of people to thank. I hope I included all of them. From the first part for the Iceland expedition, our Feldspar team, funding from a variety of different sources, the headmistress of Foscoli, which the Icelandic person in the audience can tell me how to pronounce her name, the NASA Nordic Astrobiology Summer School that I mentioned. Google it if you're interested in learning about astrobiology or pursuing that as a career. The Planetary Science and Technology from Analog Research Program, P-STAR, that's what just gave us some funding to go back. And then for the Titan lab work, Rob, Twan, Mike, Matthew, a bunch of people and funding, and thank you all for your time. So I'd love to take your questions. There is a mic since this is being broadcast, so we can all hear your questions, not just me. And I'll try to remember to repeat them out loud too, if we need to. Go ahead.
Oh, hi. I was wondering, you mentioned that there's some sort of fungus that could survive on Mars?
Oh, this is cool. Okay. Now, I don't know exactly the authors of this paper, but there's this lichen that they found in Antarctica. Now, lichen it's a combination of a fungus and a cyanobacteria that live in this symbiotic relationship. They sort of need each other to survive. And the group took that lichen, and then they put it into a Mars chamber. And they put it, some of it sort of under a rock, kind of shielded from UV radiation, and then the other one was exposed. And then they stuck it in there for, I think it was a couple of months, and it had, Mars temperature, the atmospheric conditions were correct, and the UV radiation, and it survived, which is crazy. So I suggest if you just Google something like Mars Antarctica lichen, you'll probably find the paper. But it's fascinating that, every place on Earth that we look for life, pretty much we find it. And there are plenty of conditions out there in the solar system, places like Mars, Europa, and Enceladus, where life as we know it could survive. And it's fun to be pushing those bounds and to see exactly what could be transplanted and be growing there today.
Is there anything else that we know of that could survive?
Sure. There's something called bacterial spores. These are certain types of bacteria. When they're put in stressful conditions, they go into like this hibernation state. And those spores can survive in space. They could have survived some sort of meteoritic transfer between Earth and Mars based on modeling and stuff. We found some on Earth in halite crystals that were on the order of, I believe, 10 to 100 million years old, really old. It's kind of controversial because we're not sure if they were deposited when the halite crystal formed or if they came in later. We've also found spores in amber that's like 50,000 years old. So there are things that can hang around, that can resist those conditions, those extremes, even in space, that could potentially be somewhere else.
Thank you.
Yeah, thank you. Good questions. It's great.
Yeah, did the 50 million-year-old spores, did they regenerate? They reanimate, I mean?
So the question was, did those 50 million-year-old spores regenerate? And they were regenerated in a lab, to my knowledge. So that was what was so exciting.
Do you intend to use nanotechnologies in your analogs?
Nanotechnologies in the analogs, like how so? Can you be more specific?
Well, for example, carbon nanotubes to dissuade the radiation that comes in.
Oh, sure. We have lots of plans for using sort of up-and-coming technologies like that. Some of the work that I did as a postdoc was doing microfluidic techniques, things that can take an entire lab, biologists and chemists working in a lab on a bench, and condense it down into a tiny little chip that instead of having wires that move electrons around, you have channels that move liquid around. And you can do an entire suite of organic chemical analyses on a tiny little, they call it a lab on a chip because that's what it is. So there are sorts of things like that that we are advancing the technology of Usually they have to get to a certain threshold before we'll put them on a mission. It's called a technology readiness level, or TRL. It goes from 1 to 9. Usually when you get to a 6, they start to take you seriously for planetary missions. And this microfluidic stuff is getting to that point. I'm not sure exactly about the carbon nanotube stuff, but I'm sure someone's pushing that technology along.
Thank you.
Thank you. Next victim. Yes.
At the risk of sounding a little like Darwin, some of what you were talking about for the volcanic fields, the lava fields, sounds like the way cities organically create, where first they're kind of esoterically populated, and then eventually, as they reach a certain critical mass, they start to stabilize in terms of what areas are zoned for what. Is there any, am I just making that up in my head, or does that seem to have any corollary?
That's a really interesting point. I mean, we're just starting to try and understand this, and we're doing as many measurements as we can, right? But more data is better. There could be sort of micro-environments happening, right? There could be a localized warm patch or a humid patch over here. Maybe temperature is different because some of these lava fields are still warm. We were having lunch near, not right next to, because we didn't want to contaminate, but sort of near one of our sample sites, and I remember sitting on the ground and being like, my butt's getting a little warm, and things are still geologically active in those areas, and so there could be small environments that we're not capturing yet in our techniques that we're planning on doing for future expeditions. But in terms of how cities organize and things like that, whenever you get to complex systems, right, there can be small things that lead to large changes in terms of how things are distributed and presented, and there could be some analogies there, absolutely.
And then quickly for Titan, is it possible that there could be not an environmental corollary for Titan for Earth before the oxygen catastrophe, when most life wasn't aerobic? Or I think I'm using the word right? Yeah,
yes and no. Back in Earth's early history, there was less oxygen, and there was a point in time where there was something magic turned on, and suddenly we were very oxygen rich. So in that sense, Titan can be an analog, but remember, it's much, much further away from the sun. It's a lot colder, and so certain things that would be a gas on Earth are liquid on Titan.
Okay.
So some of those analogies, you have to be careful about how you draw them. One cool fact that always gets me excited when I think about Titan is, so if you think about what stars are most common in the universe, or at least in our corner of the universe, our star is actually not the most common one, a red dwarf star. Now, if you're a planet in a stable orbit around a red dwarf, you're not really, really close. You're about 1 AU, 1 atomic or astronomical unit, which is what Earth is from our sun. You're about that far away. But red dwarfs are a lot dimmer than our star. So it turns out that if you're 1 AU away from a red dwarf, you get about the same amount of light that Titan does. So chances are, just statistically, there are probably more Titans out there than Earths. So if we discover some new chemistry that could happen that could potentially form molecules important for life or support life, that would be a really big sort of universal context discovery.
Cool. Thank you.
Thanks. Hi.
Do we have strong reasons to believe that there may be life on Titan, or was the experiment that you did, was that just to investigate hydrologic processes on the moon?
Yeah, it's a good question and it's a hard question. So thinking from an astrobiology perspective, places like this with tons of organic molecules are really exciting, right? Because what do we eat? We eat organic molecules. All life needs those type of building blocks to be able to make the structures that they use to exist, at least life as we know it, right? Now, Titan is unique because it's got these alien exotic solvents of methane and ethane, and it would be so cool, and lots of scientists are working on theories of what kind of life could exist in these places. But you have to remember that these are very cold places. Now, when things are cold, not a lot of stuff dissolves, right? We've done some experiments that demonstrate that. And life, at least as we know it, needs large molecules in order to store and transfer information, like your DNA, like your proteins. Those molecules have to be pretty big so they can store information and store function, so your proteins can bend and fold in certain ways to do certain processes that are important for your cells to work properly. When it comes to methane and ethane, not a lot of large molecules can dissolve. It's just too cold, and it's not as good of a solvent as water. Now, that doesn't mean that life couldn't exist in a place like this. It just means it's harder for us to come up with some good examples of life, at least with our Earth hat on. thinking like we do, that as life as we know it. But there are other solvents too, not just those that could exist out there in the solar or in the universe. Ammonia is a good example that can host all sorts of really interesting molecules. CO2, supercritical CO2, if it's under pressure, can dissolve things just as well as water can. So there are other exotic forms of liquids that could potentially host life elsewhere in the universe.
I think I remember hearing or reading about it somewhere that like Titan's atmosphere, I guess it's nitrogen and a lot of methane, came from like the decomposition of ammonia or something like that?
There's an equilibrium between nitrogen and ammonia that happens. So nitrogen N2, ammonia NH3. There's a relationship between those two molecules, and you can actually use that to sort of date how old things are. So there, places like Titan, we can look at that and study that. But in terms of like the exact age, I'm not very familiar with that.
Thank you.
Yeah.
Thank you very much.
Go ahead and use the mic, please, if you don't mind. I can also repeat your question, but.
Thank you. So we are talking about organic material and life. How do we go from there, and is there a definition of life for this project?
Okay. We astrobiologists, biologists, it's hard to agree on a definition of life. One of the things that we'll look for, at least when we're looking for places where water is a liquid, is we'll look for life as we know it because it's using the same solvent, and it's probably going to use a lot of the same atoms. If you just look at the abundances of elements in the universe, right? Hydrogen, really abundant. There's a ton of hydrogen. Next, there's helium. And then there are the lighter elements, carbon, hydrogen, nitrogen, the things we use. Now, which Star Trek episode is the one that had the silicon-based life? Thank you. Was it? Nice. So it's not to say things like that couldn't exist, it's just silicon is a lot less abundant than carbon. And as a life form, you want to use things that are common around you, right? Because you don't want to be starved for nutrients or building blocks of things that you need in your molecular machinery to continue to exist. So most likely, life that we find is probably going to be composed of some of those more common elements. Now, they may be combined in ways that are very alien and different. And here at JPL and other places, when we're starting to think about what the next life detection experiment should be, we're keeping that in mind. We're trying to think as broadly as we can. But we're still going to be looking for patterns of life that may contain these molecules. Did that answer your question? Sorta? Kinda?
I just had a hard time going from molecule to life.
Going from molecules to life. Okay, well, amino acids are present in meteorites. Lots of meteorites. Life didn't put them there. They formed naturally from things that were available in the solar system, in the universe. Things that reacted because they were in gas clouds that got some energy from radiation from a star nearby or something like that. So these molecules form in the universe, and when they get concentrated on a place like Earth, where they're in an environment where they could potentially react and interact, then eventually, at least we have one data point that says that leads to life. Now, what was I going to say? I was going to say something else. Yeah, so on Titan, we again have these molecules, and we found that there's something you can make in the lab here called tholin, which is our best guess as to that sort of organic muck that's on the surface, the sort of beautiful brownish, orangish stuff. Carl Sagan coined the term. It comes from the Greek tholos, which means muddy or not clear. Which is kind of fun because we don't know what it's made of, and it's sort of muddy looking. But when you put that in water, it automatically makes amino acids, like right there, spontaneously. So all you would need was liquid water and just sit and wait, and you're making some prebiotic molecules that could form life. So it's a chemistry question, I think, more than anything else. If you've got the stuff there and the chemical conditions are right, you're going to form some interesting stuff, and hopefully, maybe, life one day.
I'd like to understand why there's a variation in these life forms in the area you're measuring a little more thoroughly.
We would too.
Well, it seems to me one way would be just the transport of material, which must happen. It comes from somewhere else when it originally ends up on these fresh lava fields. And on the other hand, the life forms themselves may be propagating living, dying, certainly they can die there. Whether they live and grow and so on is another way the concentrations can vary. How much do you understand about what causes the variations?
We don't understand much about what causes those variations, and it's a point of consternation that I really want to know what's going on. So we've brought as many sort of techniques that we can bring to bear. We're bringing mineralogy experiments, a few things that I didn't get a chance to talk about today. We're measuring temperature, relative humidity, things like that, and we're trying to expand, not just looking at the general kingdoms of life that's there, but looking more specifically. Then it becomes a problem of manpower, that our team isn't quite large enough to be able to address that entire sort of area, sample space of parameters, right, that we can look for. So we're trying to do as much as we can in the field when we're there. Certain things that are transient, the amount of water in the sample, the amount of life that's there. We'll look at that right away. But then we're bringing our samples back to do the mineralogy, to do things that now we can take our time and study in more detail. And so we're looking for these correlations. Like I said, if there are microenvironments present, or if maybe some hiker, came along and accidentally kicked our sample site. I mean, these places, we're only there for a few weeks out of the year, and who knows what else is going on when we're not there. In fact, some of our sites from FIM from 2013, when we came back in 2015, Iceland had a really wet winter, and some of them had snow on them. And so we weren't able to use those sites again. And in a way, we didn't want to, because then that's too wet. That's not really like Mars anymore. So we're trying to be better about selecting sites in sort of lower altitudes that won't get a chance to potentially form as much snow and other things like that as well. But it's a hard problem. And we're hoping that some of the information, if we're able to glean an idea of, okay, well, you, Mars rover, go over here, don't go over there, because it has X parameter. We're hoping that we'll be able to do that for the next Mars mission. Any other questions?
Yeah, what evidence is there about the duration of existence of oceans on Mars and the types of things dissolved in the oceans there that you could use Earth as an analog for in terms of how long it took life to develop in the oceans on Earth? Do you have anything to say about the likelihood that it could have happened on Mars?
Okay, so
what you know so far.
So the question was about oceans on Mars, how long have they existed, what could have been dissolved in them, and how can we use analogs on Earth to address that? And that's a very good question. Our understanding right now is that Mars used to be a very warm and wet place, had water or liquid water on its surface for extended periods of time. There's evidence that the pH of that water wasn't as extreme as we originally thought, so it may be more conducive to life. There were probably a lot of salts dissolved in that too, but there are lots of salts in our ocean waters as well. So I think there's a large sample space there in terms of what concentrations of things could be present, that there are lots of scientists working in the lab to try to simulate that. We're interested more on, okay, now life has had a chance to get a foothold in one of these volcanic areas on Mars, and those are the places that we're sending our rovers to now, or our sampling planetary missions there. So that's where we want to try to get a handle on what kind of life may still be there, what is its distribution, and if something looks the same everywhere, is it really the same everywhere? And we're finding no, so. But it's a good question. There are a lot of labs that are doing all sorts of cool work to try to address what Mars used to be like.
Okay, well, we hear about life being here, life being there, and found over here and not there. But what's your formula on how is life going to form? Into what?
Well,
have you ever thought of that? Who
yes.
does that?
There are some people that do that.
It just happens by accident? Isn't there a thought out there somewhere?
Oh, sure.
Where is it?
There are a lot of people working on the origin of life, and it depends on who you talk to. There are some people who believe that life originated in some type of a warm pool on the surface, where organics were able to concentrate, and then you had energy sources from the atmosphere and things raining down in. There's another camp that believes that life may have originated in hydrothermal vents down at the ocean floor. Now, from an astrobiology perspective, looking out in the rest of the solar system, this is really exciting because we have places like Europa and Enceladus that have pretty good evidence that they may have hydrothermal activity down underneath their ice shell in their oceans. And so if life formed here in those kind of conditions, what are the chances that life could also be present in one of these places? So in terms of the exact molecular reactions that happen, that depends on a lot of different factors. There's certain associations that are favored in certain temperature regimes, under certain pHs, certain redox gradients. If it's oxidized or reducing, certain reactions will be favored and they'll form certain things. There are a lot of experiments going on to understand exactly what, if you have a very acidic pH and you have certain concentrations of things and it's reducing, what forms, what spontaneously forms, and what degrades, and sort of what things can you make, and then people looking in other conditions as well. So all I can say is there's a lot going on, and when someone makes life in the lab, you will definitely hear about it.
Yeah, but it's just like, let's say a computer itself represents life. And you turn it on and there it is, and all of a sudden you see a bunch of things on it. Well, it just doesn't happen like that. There has to be some kind of formula to point us to just exactly what direction life is going to go into. For an example, is this going to be a fish or a bear or what? Or a human being?
I think part of that formula is chemistry, right? It's entropy, it's randomness, it's these fundamental physical parameters that define our universe. Moving things from order to disorder, that type of thing. Energy gradients, right? Moving things from a higher energy molecule, something that, say, has high energy bonds in it, to lower energy bonds, and gaining energy from that. Essentially, that's all life is catalysis. It's getting over that activation energy barrier to get to a lower energetic state and using that energy as expressed as life. That's what we all do. Right? We eat food and then we breathe out CO2 and we give off other things too. And so if there was a formula, someone, it doesn't mean that there doesn't exist one doesn't exist, but it is complicated. We're very complex systems.
Well, life is the motion of these items. The question is just where is this motion going to and how is it going to form? And of course, you can't answer me that question. Thank you.
Thank you. It's always good to have these conversations.
I have another strange but quick question.
Okay. You say quick, but we'll see.
Let's see. I believe it was back like in May 10th when Mercury was crossing across the sun.
Yeah, the transit.
The transit,
Good stuff.
yeah. I'm not sure if this article is correct and the video is what I saw, but this amateur astronomer by the name of Paul Cox, I believe, he was showing the broadcast live through a solar telescope. I don't know if it was the Sleuth system or what. And you could see, you mentioned red dwarf, so that's why I had to bring it up. He saw the sun and then Mercury was crossing, and next to it was another sun. And there's been a lot of talk about this. And he explained that was our red dwarf, that we belong to a binary system. And yeah, you could see it clearly. And there's a lot of talk about this, and I've seen pictures of it like in an old 1930s Britannica encyclopedias explaining that we're a part of a binary system.
So we're like Tatooine. Cool.
So do you know anything about this?
Actually, binary star systems are a lot more common than most people think. I don't know the exact percentage, but it's a significant percentage of star systems that are out there. I hadn't heard anything about this.
Okay.
It's interesting. I think in terms of all of the mission planning, mission design that we do, when we plan trajectories for, say, Cassini to get out to the Saturn system, and we're planning for this new Europa mission that's going to go orbit around Jupiter and visit Europa, we have to plan those maneuvers very carefully. And it seems to me if there was something like that there, that would affect our spacecraft. But we have to do orbit trim maneuvers. We have to modify things all the time. We have to check for Cassini, which is right over there. Whenever we're getting close to Titan to do a slingshot around to continue to do some analyses of the Saturn system, we're always checking the positions of the other moons. It's called the ephemeris, to make sure that we are where we think we are and they are where we think they are. And sometimes we have to do corrections. So
All right, thank you.
Yeah.
Just thought I'd ask.
Any other questions? All right, well, oh, good. We've got one from the web. Let's see. BKPR1 asks, have you seen biomats on Earth? Is there anything in your research that says biomats could have formed on Mars? Good question. Okay, so we see biomats in a lot of places on Earth. Now, I'm not a geochemist, so I can't speak about them in any detail, but I know these microbial mats form, and we also see evidence of them in the geological record, too. There are things like biovermiculation is one thing where you see patterns, or fossils, fossilized microbial mats. And there are theories that we may see something like this on Mars. These kind of communities are extensive. We see them in conditions that we may consider to be extreme. And so one of our goals with Curiosity and with a lot of other future missions is to try to look for this kind of evidence in some of these layered rocks that we see. So good question. We see them here. We're hoping to see them elsewhere. Who knows what the equivalent of a biomat would be on Titan? I don't even know how we would look for that, but that would be an interesting thought exercise to do. Well, I appreciate you guys coming out and staying late, and I will hang around if you have more questions, but continue to ask questions, and remember that even your backyard could be an analog environment for somewhere else. So, thank you.